So I would start with the panelists. Do you have questions you'd like to ask of each other or stimulate some discussion here? Oh, push the...
I'd like to ask my fellow panelists whether any of us have presented data that contradicts the data that you presented. In other words, have we come at it from many different angles and reached conclusions that are consistent, or have we tried to set up a problem that we need to resolve?
Low glucose levels, I think. Go ahead. Anyone have any?
Well, I was really struck today about how all of us had similarities in our data, I think. I clearly saw a lot in what Barbara just talked about, and I hadn't really thought about things the way that you presented them, but I definitely think that what I see is definitely relevant, or what you were talking about is very relevant to what we're seeing. And I felt the same way about the adipocyte talk earlier today. And of course, Bruce's data, there's a lot of similarities there as well. So I think we have a lot in common.
Great.
Let me comment a little bit. I think the problem is that there are many targets for the pollutants. I mean, everyone works on some targets, maybe inflammation, the estrogen receptor, the energy, the pancreas, the adipocyte, whatever. And I think what we lack until now is, do we have so many real targets, or is there somehow, we are dealing with a few targets that are influencing different things, and depending on the experimental setting, you see something and the other one sees something a little bit different. It's not clear to me. I mean, I thought of that when listening to Scott's talk, and in Tox 21 you have so many pathways that you are testing, and sometimes it doesn't fit with the biological experiment that you do afterwards. So the question is, are we really looking at all the relevant pathways, or how many are still lacking, and how to do better? I mean, just increasing the pathways or combining pathways, trying to see, when something doesn't work with one pathway stimulated but may work with 3 pathways stimulated at the same time, etc. I mean.
Go ahead. Yeah.
So I think we need a systems biologist among us, and I think I was trying to, in a sort of simplified, naive way, get at that the changes that occur in the blood in response to an attack on any of these organs is going to be communicated to all of the systems, and so that we can actually study these things in any cell, because I think they're at least many things are generating reactive oxygen species, and that's changing the redox system in the whole cell. And for each cell, it's a very cell-specific phenomenon. We've shown that in the beta cell, it affects insulin secretion. In the fat cell, it affects lipid turnover, not specifically lipolysis or lipid synthesis, but both of them. And in liver, it affects glucose production. So I'm kind of bet that there are some general signaling molecules that are being generated by these obesogens that are affecting many cells, because these are not simple one-target diseases. We haven't succeeded identifying, and there have been many targets identified. None of them have worked.
Okay, so a question from the webcast. This is from Christy Poulin, and it's for Scott, based on his presentation. So she asked, you showed that the chemical enrichment assays picked up pathways you might expect for obesogens. Were there any pathways that you anticipated to be in their enrichment, for example, those from the expert analysis, that were not in your chemical neighbors?
So the answer is yes. And the way the enrichment analysis works, it's actually a rank list, and I showed you the top 15 or so. As you go down, you start seeing other things come up. One of the things about the chemical enrichment analysis, or the Sentinel chemical analysis, is it goes after one specific mechanism typically, because you pick a prototype chemical, right? So I picked rosiglitazone, which is a well-known PPAR gamma activator. But there are, as we're pointing out, there are many different ways of getting to obesity. Whereas with the ToxPi approach, we take into account all the assays that the experts have recommended, and they come from these assays are diverse. And so you get a different set of chemicals because you're looking at a diversity of assays as opposed to using just correlation in biological activity with one chemical with one very specific or a pattern of activity. So it's not surprising to me that there would not be specific overlap or specific relationships between the one analysis versus the other analysis.
Okay, and then another question from the webcast. So this is from Nick Durander, who is also a presenter for tomorrow, who's not able to be here. So he asks, are there any thoughts, and it relates to tomorrow too, are there any thoughts about what could be done effectively to reduce potential contribution of these chemicals to obesity, and have you seen any treatment data in animals?
I don't know about treatment data, but certainly prevention has got to be the #1 way to deal with these kinds of chemicals. Avoid them as much as you're able.
If inflammation is important, which is, I mean, it's a sort of it looks like, Ross in many respects, because I'm not sure it's the single thing that explains everything, but it's a sort of cofactor of many different diseases and conditions. And, well, I mean, there's a possibility to use anti-inflammatory, but that's, I mean, how long? I mean, you cannot use those all the time. So I think nothing equals really prevention in terms of public health issue.
I think the biggest challenge we have is how do we communicate these findings? How do we get the power that we need to convince anyone to make a change? That I think, that's to me the most frustrating thing. That how do you change our nutritional guidelines? How do you change the chemicals that are going into growing our plants, growing our animals? Who's going to do that?
We'll just let that question stand. I thought I had that, in one sense, bring us back to the clinical picture here. And a lot of what we've done is sort of treat this as a single disease. And my question to you is obesity if we treat it as a disease, is it a really different diseases? I mean, you have diabetes, we have some of these others. What is our evidence that really it's all multifactorial, or are there components, whether it's from the genetic side or the development, because you started at an early age with exposure and then it's transferred through the generations? Are there patterns out there that we need to pay more attention to? So that's kind of the one question. Then the other is at various levels of obesity. I mean, we tend to talk about obesity, but it's really from perhaps even normal weight all the way up, and we treat it as a linear regression, sort of. And in normal weight, does it really matter if you're 103 or 104 pounds? But we treat it statistically as if it's continuous. Do we see in your studies that at different levels of, call it mass or weight on your animals, you see a different impact by the different chemicals? So it's kind of like we have a detox mechanism we haven't talked about that is actually, we're saturating it now as you move from being overweight to obese to, morbidly obese. Do different exposures have a different impact at different levels that our bodies are, as it were, as Barbara said, leaking your energy now because you're taking in too much? Any thoughts? I'm busy taking up the hour
I'm
here.
not in
but-
the clinical business, but I mean, the clinicians that I know, I mean, they clearly think that there are different diseases. I mean, it's, you have, and they don't understand all the time why. I mean, but clearly, the simple difference is that some obese individuals are not sick in the sense that they don't, metabolically at least, I mean, or they don't have these, they have a very good, they have no diabetes, they have no metabolic syndrome, and they are fine in this respect. I mean, now they may have obviously other problems with joints and, of course, I mean, other, physical problem. But in terms of metabolism, they are doing really fine. And other obese individuals, even if they have less BMI, a smaller BMI, still have more metabolic. I mean, those are clearly very distinct for the clinicians. So this, at least, to this difference.
Yes.
There was that paper several years ago, New England Journal of Medicine, that looked at adiponectin levels in a group of young boys in India, and those who had higher levels of adiponectin, even though they attained higher BMI, were far less likely to develop insulin resistance and type 2 diabetes. So that sort of goes along with there's, an elevated BMI, as Bruce's picture suggests. Arnold may be at risk for certain health outcomes, but not specifically for type 2 diabetes, unless he was juicing. But that, so that not obesity is all the same, the visceral adiposity, but even the adiponectin. I mean, why would these boys, I mean, they had different trajectories despite starting all at the same place. Those with the high adiponectin, again, did not develop the metabolic consequences associated with obesity. And our data that we presented a little bit this morning would suggest that some of these exposures may have different expressions, whether one has a normal BMI or one has an elevated BMI. So that does beg the question, and perhaps some of the discussions today would sort of corroborate that maybe it's through inflammation or redox, or perhaps they're both the same part of the elephant.
Yeah, go ahead.
I just want to make a comment that when I say Ross, I'm probably saying the same thing as most people mean when they say inflammation.
Yeah.
Because, what do you mean? We use these terms that are undefined. What is inflammation? Inflammation is happening in a fat cell, and yet nothing ever causes a loss of fat cells. When the same inflammation happens in a beta cell, you have type 1 diabetes.
Yeah.
I just wanted to add something to the discussion, which I feel kind of started from the neck down so far. And the only one who has really mentioned it is Scott, when you showed, these are the many pathways that are targets where we need to look for obesogens. What do they affect? And one of them was behavior. And so I'm wondering, we've been focusing very much on the adipocyte, and of course, Barbara is the one who clearly links it, saying, primary hyperinsulinemia could drive hunger, and we eat for the insulin. Eventually, we become insulin resistant because we manage to gain weight. But what about a direct effect on the brain?
So when we started off doing some of the modeling, one of the first things I did was go to basically a pharmacy side effects book, and I said, what chemicals, what drugs are known to cause obesity, weight gain? And 90% of them are neuroactive substances. And you're very much correct, but I guess getting at the behavioral side of things is quite challenging. Rodents aren't quite as smart as us. And so getting behavioral characterization is somewhat challenging. But I agree. So one of the things I wanted to point out, along the lines of the diversity of possible effects, is what we need is a set of prototype chemicals that we can mine, that we can look at to see, okay, You know, we've got these over here affect neurological function, these affect adipocyte function, these affect, and start digging through those as prototypes and mechanistically and seeing what other chemicals behave like those chemicals, kind of like what you saw in the correlation analysis, the ladder analysis.
You probably have enough of those to keep you busy for a while, I would say.
Well, I think about the brain a lot. I'm trained as a neuroendocrinologist, and I think, particularly in our model, that the timing of the exposure is really the time when those metabolic pathways, both for food intake and metabolism, are developing in the brain. So I'm very convinced that bisphenol A is probably having effects, and we know for bisphenol A anyway that the brain is very sensitive. The developing brain is very sensitive to bisphenol A. We know it changes sexual differentiation of the brain. We've seen reports of changes in hippocampus, changes in cortical development, changes in neurotransmitter development, and actually in the metabolic fingerprinting we've been doing on our animals, we do see at postnatal day 2 differences in neurotransmitters in the brain. So I am very certain that one of the major targets will be, or is, the brain. We just have to figure out how to figure it out. I mean, Desai has done some interesting work showing us that certain neurons are more abundant than other neurons, et cetera. So that's a start. But these are very complex pathways, and actually, they're very complex pathways that still aren't completely worked out. If they were, I think the drug companies would be doing really well at getting us a drug that works for weight loss. But
So Dave Fukuzawa, a roundtable member. I'd like to go back to Barbara's original question, maybe tweak it a little bit. And this was a question I kind of asked Bruce at the break. I mean, all of you are basically challenging the orthodoxy, which you talked about, and all of you talked about, you know, that it's an energy balance question. And I think you all, I mean, there's a great concurrence, and I guess I was wondering, what have you heard as you've tried to make the case for this perspective on obesity versus the traditional one? And, you know, what is your sense about what needs to happen to sort of change the narrative about it?
So my answer to that is that I'm impressed with the fact that every tissue that we've looked at is affected by the same sorts of things, but the outcome is totally different. And this, to me, makes a huge amount of sense. That there are, I mean, the way systems work and the way Mother Nature works is that the same sort of techniques are used in every cell of the body to achieve particular things. And so, for example, all of the cofactors that are used for all of the enzymatic reactions that are happening in every single cell, they're using the same techniques, they're using the same kinds of mechanisms. And so when something changes that has a big change in one tissue, I think we can expect that it's going to be happening everywhere. And I don't mention the brain because I don't know how to study it. But if I did, I would focus there. And, so I think that the same ROS-generating chemicals that are affecting insulin secretion and, so forth and so on are probably affecting satiety and food, nutrient sensing. And I was going to ask, along those lines, the one thing I think back on, in the olden days when we used to, didn't have these nice model animals, we used to feed animals gold thioglucose or what is MSG to cause, in a tasty diet to cause obesity. And both of these are acting by screwing up the ability of parts of the brain to sense sufficiency. So has anyone looked to see whether any of these obesogens have a direct effect on the nuclei that are involved, for example, in those effects? I don't know the answer.
Well,
So I showed you some that
yes.
data this morning from
Yes, from
the
NPY.
Michael Ross group, where they showed that BPA exposure during development increased food intake, and they could correlate that with changes in the neuropeptide Y neurons, which cause you to, I can never get that right.
It causes you to eat.
Causes you to eat, and decrease in the number of the satiety neurons, the POMC neurons. So there's starting to be some people looking at that part. But I'd like to just continue this, because the one place where people aren't looking is on the other side, in the hedonic pathway, which is the food cravings kind of thing. And a lot of people think overweight it's emotional eating. It's like a food addiction. And there's a lot of data on addiction and changes in dopamine receptors in the brain and all of that. And I think that's a whole field that people doing obesogen research need to get up to speed on, and to look for effects in those areas.
So Mike Gorin at USC is someone who's working in that area. If you remember, one of his postdocs spoke at the Gordon Conference about 4 years ago about that, about the effect of chemicals on hedonic pathways and putting people in fMRIs and really investigating that. So at least one person's doing it.
I should point out that those agents that cause or drive obesity in humans tend to be antipsychotic medication that target the dopamine receptor and serotonergic receptor systems.
And we do have some data from, I can't remember the lab right now, suggesting that bisphenol A, early exposure to bisphenol A does change dopamine in the brain. So, and that would also go along with all the reports of hyperactivity, because that could also be caused by changes in the dopaminergic system.
Yuan Xiangpan from University of Illinois. I'd like to, continue on Barbara's discussion on the environmental agent actually in circulation affect potentially every tissue or cell types. The reason we're not seeing every tissue respond the same way because every tissue have different epigenomes, so that even the same redox going to the tissue have the same histomodifier. The effect will be different because epigenome different.
I don't do epigenetics, but it certainly makes a lot of sense to me that that is yet another factor that has to play in, and it's sort of, to me, in the same category as the microbiome. And I think in all of these things that we know there are differences, it's really important to know whether the difference is a cause or a consequence. We don't really spend a lot of time there. We tend to find a difference and then focus on it.
Okay, so another question from the webcast, and this is Nick Duranda again. He wanted to ask the speakers to comment on if their research has shown any of the chemicals crossing the blood-brain barrier.
Well, we know for bisphenol A, for example, particularly during development, it can easily cross the blood-brain barrier. That's for certain. And it's not protected the way the brain is protected against estrogen by things like alpha-fetoprotein. It bypasses that completely because it doesn't bind to it, and it gets into the brain very readily during development.
And at least some of the organotins are neurotoxins and must cross the blood-brain barrier.
Another issue that is important is what do they do to the blood-brain barrier, and some people are working on that, because if they alter blood-brain barrier, then they will let some other things get in, and that's another way of toxicity. So that's I think is an interesting subject. It's not, the answer is not completely there yet, but
I think that some of the monoglyceride type compounds resemble 2-AG or the endocannabinoids, and clearly they do get in.
Generally speaking, the small molecules can pass the blood-brain barrier pretty easily, and greasy molecules can pass the blood-brain barrier pretty easily. And anything amino acid-like things along those lines pass through quite easily.
That's true.
But I personally haven't looked, but generally speaking, those are the rules of the blood-brain barrier.
Bernie Goldstein, University of Pittsburgh, roundtable member. Barbara, let me follow up on the cause or consequence. I noticed when you were presenting on the reactive oxygen species, you were pretty careful about how you phrased that in terms of cause or consequence. Could you perhaps expand on that? Do you think that's really something which you can nail down as a cause? Are you sure it's not a consequence?
I'm sure it can be either. I'm absolutely positive it can be either. And you know, there are, but my point really is, you start out with a hypothesis, like insulin resistance is the problem, and if we could solve that problem, everything would be fine. And after so many years and so many dollars spent on that project and no tangible results in terms of improved health, it's time to look and see whether there's another hypothesis that might also be good, that might help us to find our way a little bit better. So I present that insulin hypersecretion is a hypothesis, and we've shown there are some ways that it can happen, but obesity as an initiating cause could also be a problem. And, you know, gold thioglucose in the brain could also be the initiating problem, because I guarantee it will cause all of the others. So that, I think that's a really important point. But if you want to solve a problem, you have to know where it begins. And we don't know that. There's no real, you know, or I guess we do know that all three of those things, for whatever number I've said, can start it. And so which one actually does, it probably varies. Like the Pima Indians have high insulin levels prior to developing obesity and diabetes. And those high insulin levels predict that they're going to go that way. So maybe, at least in that population, it is cause. But I don't know.
It's interesting. I think we spent 2 years doing glucose tolerance tests in our animals, starting at 8 weeks of age. And we never saw problems with glucose tolerance. And so we were pretty convinced there wasn't a glucose homeostasis problem until we started looking at fasting insulin levels. And we were floored that by 13 weeks of age, and we haven't looked at younger ages, we could see significant increases in insulin. So I do think that the hyperinsulinemia question or fact is a big factor in some of what we're seeing.
Just to follow up a bit, I think if some of my colleagues who are diabetologists here would argue with you as to whether the metabolic syndrome is not, their understanding of it has not led to improvements in diabetes care. You're absolutely right, of course, that it hasn't.
The improvements aren't enough. And, you know, I would argue that, too, if somebody started attacking my favorite hypothesis. But, you know, but I think this is the game we play, and I think it's an important one. And I'm going to be wrong 20 years from now, no matter what I come up with, and so are they.
Quickly ask another question for Barbara. If the sweet taste receptors were knocked down or knocked out, would the artificial sweeteners still cause hyperinsulinemia?
Yeah, I don't know. In fact, I've left that open a little bit because I haven't studied the receptors, and I know they're there. And I know they're in many places, including fat cells, which is a silly place to have sweet receptors, I think. But whether or not they are the agents that are acting, we don't know.
Other comments, questions?
Henry?
Oh, there she is. Yep.
Hi, Melissa Perry, George Washington University. So, Barbara, I was really struck by the slide where you pointed out how the nutrient composition of foods have been changing rapidly. It was just eye-opening, even though I guess we should know that, but it was just useful to be reminded. And it made me realize that we're looking at a different approach to growing a variety of different crops to include pesticide coating and treatment on seeds.
Yeah.
So, believe it or not, between 2004 and today, we have seen a takeover of corn and soybean, with about 90% of the seed that's planted in this country of corn seed being treated with neonicotinoid pesticides. And I don't think we know what that means in terms of the overall composition of corn and what it means to us in terms of ingestion, and certainly what it means with respect to environmental impacts as well. So I can't help but to pile on here to say yet another element, another competing element as to the introduction of chemicals into our food.
Exactly. And you know, as scientists, when 10,000 things have changed, how can we focus on one of them? How do you decide that it's overeating that causes all these problems and not, I mean, I didn't know, you just taught me something, but I'm not surprised because when the mineral content of peaches is measured, it's different. So you would, you know, everything has, so many things have changed, and we don't have a test system in place in our society to not let these thousands of things enter without appropriate testing. So, you know, that's one of the things I think that could be done to make things a lot better. And, you know, certainly in the food supply, everything that's been added has been tested, and has been tested for long periods of time in rodents, at least, to make sure that it doesn't cause tumors, birth defects, whatever. But we can't get that information on the body weights. So, you know, that we should be able to get that. Nothing should go into our food supply without that testing, including the most prevalent diseases that are occurring in society today. But I have no idea how we would handle that. I think we need politicians on this.
Since you brought that up, and since we're here at NIEHS, I think that this policy of allowing companies who produce a product to be the ones who test it for safety is a bad one.
All right.
Yeah. No, I didn't say it was your policy. I think that this is perhaps the best place that that kind of testing should be done.
I'm not sure we can affect any change in our government. But we can try.
Yeah, I'll add a question for Chris at the end there. Where do we stand on phase 4, phase 5 of testing? I mean, when do you see the end of this?
So right now, all I can see is phase 3. One step at a time. We're learning our lessons. Bruce has pointed out we are learning our lessons. We are moving forward. The data that Bruce was referring to was phase 1, and we've since learned. We've learned quite a bit in what goes wrong and why things go wrong, and we also realized that we need to add a number of variables, complexity being one of them. And so in phase 3, we are planning on doing a variety of different cell types, so cellular contexts, looking at high-dimensional screening, so transcriptomics, in-dose response, looking at image analysis of the cells along the lines of looking at, say, lipid accumulation. What do I see it going after that? I think we're going with human on a chip, probably. That's going to be the next evolution of it, where you start integrating different cell types into microfluidic systems and things along those lines. That's a ways away. DARPA's working on that now. We're seeing some positive outcomes from that stuff, but it's got a ways to go to particularly make it to the scale that we want to use, which is 10,000 chemicals, or if you look at REACH, 120,000 chemicals. So quite large.
So how does your data compare to the pharmaceutical industry generated data? And
so if we've never really had access to pharmaceutical high throughput screening data, just the same way that we're slowly getting access through ToxRefDB to some of the proprietary studies from the pesticide manufacturers and things along those lines. So it's really hard to compare. I do have a fellow that works for me that worked at GSK for a while, and he says our scale is very small compared from a library standpoint. They screen millions of compounds. We're in the range of 10,000. Their assays tend to be, at this point, very congruent with ours. A lot of what they use is what we used. We borrowed from them. The next evolution of this is going to be those high-dimensional screens, and there's fewer of those going on. There's more cellular apical endpoints and things along those lines.
Okay. Thank you. Barbara, you started to speak, I think. No.
I would just point out that of those millions and tens of millions of compounds that they screen, there's only a few hundred different structures. My chemist friends say, you have to be the most incompetent chemist in the world to screen 2 million chemicals.
Yes, that's one of the points I made in my talk. Our chemical diversity is very broad. It's sparse from an individual chemical standpoint, but very broad from a structural variation and property standpoint.
Go ahead.
So, Yuan Xiangpan from University of Illinois, in your phase 3, will you be able to look, more into the new released NIH epigenome roadmap in 111 tissues to see whether the more universal response than just the direct response from the receptor approach you have right now?
Yes, and that is the plan. Actually, we have one of the people in the group that I work with is working on mining massive amounts of baseline gene expression data across thousands of different cell types to identify a diversity set of cells that broadly represent biological space. And so, yes, we'll be able to ask that question specifically and address that question that you have, saying, is there that consistency across cell types? And I would make the point that I've been saying nature is very parsimonious for a very, very long time. And so, yes, nature likes to borrow genes to do a variety of different processes, so it would not be surprising to me if Barbara was correct.
Hi, Al McCartland from EPA and a roundtable member. I am not a health scientist, I'm an economist, but I'm trying to do some homework and get a data point for tomorrow's discussion. And that is, most of the work here has obviously been with animals and animal models. And what would it take to come up with a dose-response curve that we could apply to subpopulations in the United States? Because we talked about where would this go in a sort of regulatory arena. That might be sufficient or a necessary condition, I'm not sure.
Well, we certainly have access to a lot of human tissue, and we can look at, in detail, the precise time course, dose response, and targets within any given tissue. And I think that there's increasing emphasis on using human tissue or humanized tissues.
So I was part of the working groups, along with Robert and other people, that was working on the cost of endocrine disruptors. It was focused on the European Union because that's who had legislation coming up. But one of the surprising and sobering things for me and for many people was, despite the huge volume of data that we have, how little of it we could use for the exercise of trying to figure out what's the attributable fraction of disease burden due to chemicals. We need good longitudinal studies. We need a lot more biomonitoring than we have. And those two things would go a long way towards being able to link I would argue, pretty good laboratory animal studies with human outcomes.
Can I just make a very simple comment? Just being a toxicologist, one of the things I will tell you is just doing the chemistry workup and making sure your chemical's stable is absolutely critical for regulatory decision making, and that chemistry can be quite expensive. There's a reason why NTP studies cost what they do.
Scott, do you have any sense of how many people have, I looked at your website there, but how many people have downloaded the data and how many grad students are? It's a tremendous database, and I just haven't got the sense that people are rushing to start to analyze it like they are with the NHANES biomonitoring data. There's publications coming out all the time. Now, maybe I'm missing the literature, but
so a lot of it is being done in the computational tox side of things. The EPA has been working over their data set quite extensively. There's been a number of publications that have come out of Tox 21, primarily focused on individual assays. There is, because of legislative reasons in Europe, there is a very big interest in our data, and they're actually, most of them that I've talked to, they have a tremendous number of great informaticists over there. They're exceedingly grateful for the data, because that's what feeds all informaticists is data, and it's to be able to generate it. So yeah, so there is a, I keep dropping off here, I'm sorry. I'm trying to.
We put you away on the end.
Yeah, there is a number of people that are using it. They're not using it for definitive decision making at this point.
Yeah.
And there are reasons for that. Bruce has raised some of them, and we're working through the chemistry because we have to make sure our chemicals are stable in those wells. You know, they sit on a machine.
Yeah.
And so there's that issue too. And so there's things we're working through, and we'll get there. It's just, you know, from a regulatory standpoint, we're not quite there yet.
Well, it sounds like something ought to be promoted to be worked with, right? So, yeah. Oh, boy.
Go ahead. Linda Birnbaum. So just from that kind of question issue, NIEHS and EPA are going to be holding a workshop this summer. We're still looking to identify the time, but to look at some of the issues of what do the regulators really need in order to use some of this high throughput rapid screening. And I should say not only high throughput, but mid throughput and so on in decision making.
I mean, you know, and just to add to, I think, Al's question, just to be really clear, I mean, that what his office at EPA has to do is to actually try to quantify the benefits to health of a regulation by things such as, say, preventing cases of type 2 diabetes and or strokes and or, you know, other impacts, lost days from work, whatever. And so this is very challenging at this point in time, I think, because I think we're seeing a lot of indications of health effects that are not necessarily tied to, and maybe there needs to be some collaboration to do this, to link to, you know, what the way the clinicians look at these things in terms of the diagnoses of disease and the outcomes, the adverse outcomes from the diseases.
Bruce, can you tell a little more about the project that you were involved in, which some of the results from that are being published this week? The one that you referred to briefly about the economization of a variety of different kinds of health endpoints and for the EU.
Well,
the impact of endocrine disruption.
How to say that? There were 5 working groups, picked experts picked in different areas, and we were charged with identifying high quality studies that met criteria, which were presented to us by Leo. I always forget Leo's last name.
Trasandi.
Leo Trasandi. And we worked through lots and lots of data and chemicals, and the process was to see which studies, first of all, which were longitudinal. So it was decided that the only studies that could be used for attributable burden were longitudinal studies, and that preferably had multiple points of follow-up. And there were, so that reduced most of the studies. Good quality animal studies, preferably using the same chemicals and similar dosing or similar exposure scenarios. We just tried to put together how confident were we in the quality of the data, and there were various factors applied. And I think that the end result of the exercise was extremely conservative. So I would say, from my perspective, the numbers that are going to come out are going to be an absolute floor for the cost. We only ended up considering a very few chemicals, many of them old chemicals like DDE and PCBs, because those were were where the best data were. And I think are going to represent only a tiny fraction of the chemicals that are causing disease burden. I don't know if that clarified it much, maybe. Rivera, you want? Or actually Jerry, who was heavily involved in this. Jerry's hiding.
No, I think you covered it as much as we can say, because all the results are embargoed until Wednesday morning.
Thursday.
Thursday. It's going to be a big number, but not as big as I think it really
Thursday
is.
morning.
Thursday morning.
Thursday morning.
Yeah.
Say it again.
So the most important component, I think, was that it was a structured process, and there was criteria of choosing all the studies, but then even moving beyond that to do the economic cost estimates. And so it wasn't picking out of the air how to determine an attributable fraction. It was using the Delphi process to come up with those estimates.
Okay, we got one last comment or question.
Thank you very much to have that last question. So Songkyun Park from University of Michigan, and I want to ask you. So the Dr. Baruki talk about like acute versus chronic exposure, and then Dr. Rubin also mentioned like the window of susceptibility, and so prenatal, perinatal exposure, and then peripuberty exposure. So do you think like the timing is more important, or like just chronic low dose, exposure might be? So the overall the answer might be the timing, and the any specific time point might be more important. But in terms of the general population, the recommendation standpoint, the low dose chronic exposure also should be very, very important. And also in terms of this, the aging population growing up, and another window of susceptibility, such as like the just pregnancy period or menopause, might be also very important. And during that time, the metabolism, fat metabolism would change. And so I just want to ask you like your insight about this, timing versus chronic low dose exposure, or broadly this the window of susceptibility.
I think, as you said, it's important. I mean, timing. I mean, it's not the same if you have an acute high level exposure or a small chronic exposure. But I think more generally on that, we are relatively well prepared for dealing with acute exposure, because probably in the evolution that was the the most important thing to survive, to keep possibly reproduction, things like that. But whenever we adapt, detoxifying, etc., there's a cost for that. I mean, in any case, I mean, and that cost it's more in a chronic way. But as you said, the difference between acute and chronic is, of course, very important.
And I clearly, we know from the epidemiological studies that we have in adults that there is an association between the levels of bisphenol A that people have and heart disease and various other things, diabetes, hyperinsulinemia. So there are those correlations out there in the human literature. And I guess what we were trying to study was a period that we thought would be very likely to produce lifelong results, and that was during development. But I'm sure that any time that we're being exposed to these chemicals, and unfortunately with bisphenol A, it's basically all of us are being exposed all of the time. So it's just, it's there and it's having an effect, and but clearly there are certain windows of exposure that may cause a more profound effect, persistent through life, and I guess that's what we've been studying anyway.
Well, I want to thank